Plant propagation. Plant propagation is creating more plants from existing plants. Mostly we're talking about cloning, but sometimes we're not. And I'll go into those types of differences. So let's start with the first types. Seeds, the most basic way that plants reproduce themselves, they produce seeds. There's genetic recombination that's done in the process of producing the seeds. So the seeds don't always produce a plant that's exactly like the parent. That's the whole point, is for pollen and eggs to join with different genetics and form slightly new genetic variations of the species that potentially could be more adapted to the conditions. So just like animals, where we don't look humans don't look exactly like our parents, but we have similarities. The genetics have changed. That's this an analog to seeds in plants. So the seeds, you get variation. If you try to propagate all these other methods I'm going to talk about, is are cloning methods. You're taking a piece of the tissue of an existing plant and um, using a number of methods, make it grow roots and leaves and turning into a new plant. So taking cuttings, like stem cuttings can be taken. You take a piece of the stem off of an existing plant, put it into some kind of soil, or what we in propagation we call it media, and sometimes use hormones and induce it to produce roots off of that stem. And then once it has roots, it produces leaves and it can turn into a whole new plant. And now the genetics of that cutting, that new plant from that cutting, are exactly the same as the mother plant or the parent plant. So you're cloning it. And in the horticulture industry, if you're trying to produce a bunch of poinsettias or Gerbera daisies that are exactly a certain kind of color, the flower color or characteristics, and you're trying to sell those, you want to clone the exact genetics of that so that all of the new clones from that look exactly the same. You don't want to use seeds because they will vary, and people go to the store, a nursery, to buy something exactly like what they saw outside or in a magazine or at their neighbor's house. So cloning is the way. From cuttings, you can do stem cuttings. You can also take some plants respond to leaf cuttings, where you can propagate a whole new plant just from a leaf. And then also some plants respond best to cuttings from the roots, in which you induce um, stems to grow off of and more new roots too. Divisions is another form of cloning. When you have a plant that forms clumps, you can dig it up, divide it up, I mean literally cut or kind of rip it apart and turn it into more than one plant. You can increase your numbers of that plant through divisions. But they're all that clump that you're dividing up is, again, genetically, it's one organism, so when you pull it apart, they're all genetically the same. They're clones. Layering is a method where you take um, intact stems, so stems that are not cut off from a mother plant, they're not cuttings, but the branches of those that plant, that mother plant, can be bent down, like lean down to the ground and touch the ground or soil. And a lot of plants, when that's done, they will form roots off of the branch where it's touching moist soil. And then once the roots form off that branch that now has some roots halfway up the branch, you can then cut that off and it will form a new plant. So we'll go into more detail on all of these. Just wanted to give you a little bit of an outline. Grafting is another form of, it's a type of propagation. It's not really about increasing numbers, but it's about getting an organism, meaning a mature plant that has the characteristics we want, and it's adapted to your conditions. So for example, most fruit trees that you can think of are grafted. With a grafted tree, like take an avocado tree. So if you go out and look at your avocado, if you look closely, you'll see down at the bottom of the trunk of the avocado tree, there might be a little bit of a bulge or a line, a horizontal line around the trunk. That is where long ago, when that plant was first created, someone grafted a branch of a tree that produces a really good avocado, like a Hass or Bacon, You can take a branch of that and you graft it or attach it to another avocado cultivar that that has a really good adapted root system for the soil. And you cut off, you have two trees to start with, and you cut off the rootstock top and you add the top of like the Haas, for example, branch, and they will grow together. So you end up with one tree. The root system is one cultivar, which is a hardy root system, and the top, this trunk and the leaves and the fruits are coming from a different cultivar. So two different cultivars grown together in one. And again, citrus, fruit trees, apples, pears, plums, avocados, they're all grafted. The last is called tissue culture or micropropagation, where one can take little pieces of tissue um, and put them in a lab situation, grow them up on like a petri dish on certain types of hormones and nutrients, and induce that to grow more cells, and then with hormones induce it to grow a new little plant. This is becoming a very big industry for even for nursery plants. Growing them through the tissue culture method, you can get a lot of new plants in a short amount of time through tissue culture. Propagation of annuals. We're going to talk about that, and mainly it's through sexual reproduction, and that means the formation of seeds. Um, the seeds, again, are of mixed genetic diversity, and those are going to have various characteristics that aren't exactly like the parent plant, but it is still a way that we can propagate more annuals. And annuals are plants that live for just 1 year and then die. So it's different than like a big shrub or a tree where you could take a lot of branches off it and take cuttings and propagate them. With annuals, it's usually done with seeds because there's just not long enough in 1 year to take a stem off and try to grow roots off of it. Although some people have done that with things like tomatoes. With seeds, they often have some kind of germination requirements. In the natural setting, a seed would go through a winter. Maybe it needs a cold treatment that mimics a winter, or a hot treatment that mimics a summer, or a lot of seeds in the natural environment will go through the gut of an animal before they are able to germinate. If we are trying to germinate seeds in a nursery situation, we might need to treat them with acid, which mimics the acids of a stomach of an animal. Some seeds need fire before they germinate. And in general, there's two ways to treat seeds to get them to germinate. Stratification is usually like a hot or a cold treatment. Scarification is some kind of mechanical treatment, like roughening up the seed coat or cutting a little notch in the seed so it's able to take in water and gases. Saving seeds. When you're going to gather seeds and use them for propagation, you can buy seeds, of course. You can also gather seeds in the wild, and you need to make sure you're doing the timing correctly and that your technique is correct. So time it so the seeds are mature and have gone dormant already on the plant. But you're not taking them too early when they're not even mature or they haven't gone dormant. So usually green, kind of moist seeds are not fully mature. You want your seeds to mature fully and then dry out on the plant, and then you'd collect them. That means they've fully matured and they're dormant. That's the timing. Technique, you just need certain techniques so you're able to gather the seeds effectively and not lose a lot falling all over the ground off of plant, and cleaning them correctly. So when you have seeds, and usually seeds have some kind of covering or they're in some kind of case on the plant, and you need to take everything off of the plant, everything off of the seed, so just the seed is what you store. And that usually means that there's no moisture left around the seed. If you took just like an old flower head off your plant and shoved it in a Ziploc bag and thought, I've saved seeds, all the other stuff that's not a seed that's around that usually is kind of has moisture in it, and it will cause the seeds to rot. So you really want to isolate the seeds before you store them, and of course dry them out as well. So back to seed dormancy. Really, you have to allow seeds to germinate well, excuse me, seed dormancy allows the seeds to germinate only when the conditions are correct for germination and survival. So if you're a plant growing in the East Coast or colder climates like Canada and you produce seeds, um, and they in the late summer and they fall to the ground, and if those seeds, let's say you had some fall rainfall, some rains, some storms in the fall, and the seeds got wet and they took in water and they germinated in the fall, and then there's a snow, and that would kill the seedling. So seeds have a dormancy that usually keeps them dormant until conditions are good for the seed to germinate and survive. So often a lot of seeds that are like wild, native plants adapted to cold climates, they they have a dormancy that does not allow them to germinate until they've gone through a cold period. So it's like they're sensing winter's over and then they can germinate. For example, dogwood trees produce those mature seeds in the fall, but the seeds stay dormant until they've gone through a cold period, and that lets the seed know, okay, winter's over. Now, if it's warm and wet, it's not just um, a fall storm and heat wave. It's actually after winter and it's springtime, and the seed, can now germinate and grow, and I'll probably survive. A dormant seed is a viable seed that does not germinate. So that's what dormancy is. It's not a dead seed. It's still dormant, and you need to do something to break the dormancy. And that dormancy is regulated by environmental factors usually. And the thing that breaks that is having the sufficient moisture, proper temperature, warmth, oxygen in the soil, and sufficient light. All four of those things happen in the springtime, and that will break the dormancy of lots of seeds, um, and then they'll germinate. Some seeds do not need a cold treatment. They're not waiting and sensing that they need to go through a winter. Some seeds do. Some seeds need a hot treatment. Again, there's various um, ways of breaking the dormancy. When you buy seeds, like vegetable seeds in the store, a nursery store, garden store, and you put them in your garden, they are dormant. So those do not require a heat treatment or an acid treatment or a cold treatment. All they required is once they dried out, they went dormant. They went, so obviously, I mean, you might not have thought about it like this, but if you bought poppy seeds or tomato seeds at the store, they haven't been, if they weren't dormant, they would possibly be germinating inside that seed packet. So, but again, they're waiting for moisture, warmth, soil oxygen, and light. Most seeds don't require light, but some do. Germination is inhibited by the seed itself. There's also so it's external dormancy of that seed. So it's waiting for those proper environmental factors, but it also sometimes has built into the seed coat is a dormancy factor too. So if the seed, it has a hard layer around the seed that doesn't let the moisture in because it's waiting, it has a requirement for that seed coat to be degraded over time. We can use what's called scarification, where we roughen up the seed coat. You can use sandpaper or a knife or tumble them like in a rock tumbler with sand. There's a lot of ways to do it that kind of roughens and weakens the seed coat so it's able to take in water and gases. But there's also internal dormancy. Those are the ones I was talking about before, where the seed just biochemically is knows that it's the right time or not internally. Seed scarification, again, making the seed coat permeable to water and gases. That's where you are weakening the seed coat and roughening it up so that gases and water can get in. That will break dormancy of seeds that have that rough, tough seed coat. Now, a lot of seeds don't. Although, like I said, the wildflowers, and a lot of wildflowers you buy in like a nursery or vegetable seeds they don't have tough seed coats. You just need to add water and they germinate. But there are a lot of native plants and shrubs and trees do have that. Um, freeze. This scarification happens naturally. In some ways, cold temperatures or microbial activities will weaken the seed coat. in a natural situation, or seeds passing through the digestive tract of various animals are exposed to acids, which weakens the seed coat and makes them more permeable to water and gases. Then they can germinate. Then the ways that we would, so in plant propagation, we're trying to, we don't want to, wait until a coyote eats a seed and then go find its poop and then plant the seed. We got to find other ways to do it that are quicker. Like I said, we can mechanically alter it by using sandpaper or knives, some way to roughen the seed coat. Um, Sometimes seeds are really big and you have to gently crack them with a hammer to get through that really thick seed coat, like a lotus seed is like that. Sometimes hot water is a way. Soak them in hot water, and that will also weaken the seed coat. Weakening of the seed coat can be done chemically using acids. You can soak in a dilute sulfuric acid, and for each particular plant or species, you'd have to do a little research to figure out exactly the strength or concentration of the acid that you need to use and the acid that best works. So I'm not giving you those details right here. Place in the glass container and cover with sulfuric acid. Gently soak for 10 minutes or so. Various durations of treatment depending on the species. Sometimes you can just soak them in vinegar. That's an acid, it's acetic acid, and it's a very low grade acid, but it can do the trick too. So you're not dealing with really kind of dangerous acid chemicals. And then after the treatment, the seeds are removed, washed, and then they can be planted. Careful if you are actually using sulfuric acid, as it is dangerous. Other techniques to break dormancy: seed scarification, again, just repeating that definition. After scarification, the seeds should look duller, not so shiny. Like the seed coat is basically roughened up. It has lots of little holes in it now, so it'll appear, not microscopically it has holes, but it'll appear duller to you. Maybe some little cracks or fissures, but not too much so that didn't damage the embryo inside. And these, once you've scarified, usually you have to plant them right away. They don't store after that because the whole way they're able to be stored is that they've gone dormant, and you've now, with scarification, broken the dormancy. Stratification is breaking the internal dormancy. Scarification was breaking the external seed coat dormancy. Stratification is, again, usually hot or cold treatment, kind of mimicking mostly winter with the cold treatment. Hot treatment is for tropical species usually. But the dormancy um, of seeds that need to go through these temperature treatments, it's adaptive, so it's preventing those seeds from germinating when the environmental conditions aren't good. It's waiting. Types of internal dormancy: there's shallow, which disappears with dry storage. A lot of vegetable seeds display this type of dormancy, so it's not too hard. You don't need to treat the seeds in any way to overcome that kind. Again, with vegetables and wildflower mixed seeds from the nursery, usually just planting them does the trick. Seeds won't germinate until subjected to a particular duration of moist pre-chilling or moist warm periods. That's temperature stratification. Cold stratification, one of the most common here in North America. A lot of our plants are cold dormant, and you can mix the seeds with some kind of moist media and put them in the refrigerator for a period of time. So I've done this quite a bit with native plants here in the mountains of Southern California, where I used to work for the Forest Service up in Big Bear, and we would collect a lot of the seeds, and some of them would require this. So we would just take a Ziploc bag and put moist sand in it and mix the seeds that we wanted to. Sometimes, for example, acorns, although those usually don't require it. But other things, sage seeds, sometimes some manzanitas, things like that. And you put them in the fridge for 30 days, 60 days, 90 days. It depends on the species itself, what will work the best. And after that period of time, you look in the fridge and you see some of them have started to germinate, and you take them out and then you can plant them at that point. Closed container. The fridge is usually about 40 degrees Fahrenheit, and that works. That's good approximation of a winter period for most plants. You want to keep the media moist but not soaking or dripping. Again, mimicking winter, mimicking a winter soil, which usually in these types of plants, they're used to a freezing winter. So the soil in a place where it snows is not like dripping wet, it's frozen, but there's moisture in there a little bit, but not a lot. Sometimes plants, if you sow the seeds naturally in the fall, they will go through um, the winter, and the internal dormancy will be broken by this natural process of winter. That can happen too, and that's fine. You will, however, if you're doing this as like a business where someone's asked you to um, produce a bunch of species, let's say a bunch of manzanitas that need a stratification, you might not want to wait that long to a whole like 6 months of a winter, put them outside and see if they germinate. Once they germinate, pot them up. That's a lot of extra work rather than just getting them to germinate quickly. Usually 30 to 60 days is the longest you need to do it in a fridge, so you can get these plants germinated and growing in a pot much quicker. Also, if you planted them outside in the ground, it they some would be lost to like herbivores like rodents coming and eating them. So fridge is safer. Worm stratification is just similar to cold stratification. You mix the seeds with a medium, moist, store in a closed container, keep at a temperature between 68 and 86 degrees Fahrenheit, and it varies with species exactly the temperature. Um, and not much more about that. We don't deal with that too much here in the northern hemisphere. Double dormancy is species that need their seed coat, the external dormancy to be broken, and the internal dormancy to be broken. So when you're doing this, you first scarify them, that is, roughen and weaken the seed coat so the seed coat is permeable to gases and water, and then stratify them. If you did it the other way around, nothing would happen, because if you put them in if they have to have both dormancies broken, they first have to have the seed coat become more permeable for the stratification to even work. They're not going to germinate if the seed coat's, as tough as iron, because it hasn't been scarified no matter how long they've been in the fridge. After these treatments, sow the seeds under the proper environmental conditions for germination, usually put into a pot, good sterile media, and you put them in a greenhouse or outside, and voila, you've got a bunch of new plants. Again, these are seeds. They're not going to be exactly like their parents, but this is how you do seeds. Some examples, oak trees, usually oak acorns do not go dormant. If you plant them in the fall, the roots start to emerge during the winter and the shoots top part grows in the spring. But acorns of black oak germinate better if they're moist pre-chilled for 1 to 3 months. So it's not always absolute that in this case acorns can go either way. But if you're going to hedge your bet and get them faster and get more of them to germinate, you would do a cold stratification of something like a black oak. Acorns of red oaks should be planted in the fall or stratified for 1 to 3 months and then planted. And again, I'm giving you generalities. If you were really going to do this, you need to look up research references on what works best, and then talk to other nurseries if they're willing to share that information with you about how to do it. Because if someone asks you to, produce 2,000 black oaks for a restoration project in, the Bay Area or something like that, or up in the high mountains of Southern California, you're going to want to do it, in a way that is as efficient as possible. Red buds, we have the Western red bud that grows around here. Um, they are best treated, scarified for about 30 minutes in some kind of vinegar or low concentration sulfuric acid, and then cold stratified after that. So the sulfuric acid is weakening the seed coat. That's scarification, and then you're stratifying them in the fridge. And then at that point, you can plant them and they'll do well. And they really do grow very easily once this is done. But without that, they do not. Maples, there's a variation in dormancy because there's so many different maple species. We only have a few around here. Big leaf maple is one of them. Spring maturing seeds of things like red and silver maple can be collected immediately um, and planted. I've seen it when I lived in Ohio. Maple trees would drop their seeds in in late spring, and they would fall in a lot of these big trees. The seeds would fall into the roofs of houses, and then if people didn't clean their gutters out, they would sprout, and you'd see like these little maples growing in the gutters were kind of leaves and some kind of not soil, but enough leaves kind of sat in the gutter for if they sat long enough, then the seeds could grow in there, and you saw these little maple seedlings on the edges of people's houses in their gutters. So they germinate right away. And then there's other species like southern sugar maple, where maple syrup comes from, and they need to be stratified. So it just depends on the species. Seed sowing, moisture, media, warmth. Containers are needed. You need some kind of pot or tray, which is a low, wide pot, or flats. Sometimes you can use this synthetic type of media like rock wool, or you can use natural soil. The general rule of thumb for the depth to plant a seed is about 2 times the diameter of the seed. So if you have a seed that's a 1/4 inch in diameter, you would plant it a 1/2 inch depth down into the soil. If you have a little teeny poppy seed that's really little, you would probably sprinkle it on top of the soil, and then sprinkle a little soil on top of that and then moisten it. So that's that. You need to, most seeds, like I said before, require moisture and warmth. So they need to be in a climate where it's kind of warm, or a greenhouse, and then moist. And they just stay moist until they germinate. They stay moist until they have roots where they can get moisture from the deeper parts of the soil. And they need to stay moist even when they're little seedlings too. And partial shade, partial light. They don't. A lot of seedlings need a shady, a little bit of a protected environment. It's going to make them do better. Not all, but some. Here's a picture of rock wool. This is where each of these, one plant would be planted into that. A little plant would be grown in a small pot, like a peat pot, and once it germinates and gets big enough, it could be inserted into the middle of these rock wool containers, and then these rock wool containers are set in a tray where there's moisture. So these things absorb water or nutrients, whatever you've got in your water. These rock wool pots will absorb the water and keep it around the plant. So this is a type of system that would be used in like a big production greenhouse for like cut flowers, where they'd grow them in these for a couple of years and get flowers off of the top, but be able to regulate them and not have to use soil. Another method, this is rock wool cut up into little chunks, and you can plant a small, not really small, not like a seedling, but a moderately mature plant into this, and then keep these hydrated with nutrients and water. So these are like kind of like hydroponic systems, but just varying, showing you kind of some various soil media types besides regular soil or potting soil. These actually are anthuriums. They're grown in a hothouse in Carpinteria. These are cut and sold and sent all over the world. These are very popular tropical species. You often see them red, not white. But these are all grown in those rock wool, that media that's rock wool. It's kind of like it's a volcanic material that's superheated and spun, kind of like insulation looks, before they use it for plant media. But it's a good soil substitute. And then these are kept wet, irrigated with water which has nutrients in it. This is the same company. It's called Everbloom in Carpinteria that grows over 250 different varieties of Gerbera daisies, different colors, and they're all done using rock wool and in a hydroponic system. These are not grown by seed. I just thought they were neat pictures to show you that kind of related to some a different media type used. But if you took the seeds of these flowers you see and planted them, you would get, for example, the ones in the very front that are reddish, you would not get, those seeds would not produce the same plant. They'd produce a variety of different kinds of colored flowers. And usually when people want to buy one of these red flowers, they want it to be exactly the same. So cloning would be the way to go for these to reproduce more of the plants. But again, these are grown for flowers. In-ground sowing, you can of course do that too. Use fine, soft, or friable soil, no weeds, no debris, no large particles. You can broadcast plant them, means just kind of throwing them out in a wide area, or plant them in rows. Rake in the seeds, covering them with dirt protects birds and rodents from coming to eat the seeds, or from cats smelling the seeds and digging them up. Keep them moist and warm if you can. If it's a cool climate, you need to have some kind of covering for them. When the seedlings do germinate, if you have too many, you can thin them out and then plant them in their permanent spots. So the needs of baby plants, the little seedlings, they need an irrigation system to keep them moist, or moisture is needed. They need a heat source. They need warmth. They need media. Perlite or vermiculite are two examples of artificial soil types to use. They need partial light. That's usually provided by some kind of shade cloth structure or shady area. They need protection from pests, fans, and enclosures. So having a greenhouse helps a lot protect plants from pests. And we're talking about when you're propagating plants and they're young and they're vulnerable. We're not talking about landscape plants outside. This is when you're doing propagation. They need to be protected. Fans keep the air moving. So if you have a nursery or greenhouse, you have fans to keep the air moving so the moisture doesn't sit right against the plant, the little seedling, for too long and cause rotting. Rotting is from a pest, usually from a fungus, sometimes bacteria.